Yes. Can y'all handle that?
Yes, sir.
It's going to be very, very anxiety-ridden moments for you. We did that yesterday. All right, let's talk about the cytoskeleton. That's this stuff. Cytoskeleton. Can you say cytoskeleton?
Cytoskeleton.
Golly, that was good. It has different types of proteins that make it up. They're not going to get too specific about this on the AP exam, I don't think. But you never know. It could be on a quiz, could be on a test. You need to know what it is. Oh, there we go. These are called intermediate filaments. They're called intermediate because they're not real small, they're not real big. Smaller than intermediate filaments are called microfilaments. They're a lot smaller. Here, this shows the different types of cytoskeleton that's involved in cell division. You all remember when cells are dividing? The spindle fibers go to opposite. The centrioles go to opposite sides of the cell, and they fire out spindle fibers. Do y'all remember those?
Mm-hmm.
Spindle fibers are called microtubules, and those are the bigger fibers. So you basically have microtubules, intermediate filaments, and microfilaments. Those are the three kinds of sizes that we're talking about. And they're basically proteins. So you remember what a protein is?
Yes.
Protein is a chain of amino acids all folded up. And so you take this protein and you put it with another protein and you kind of hook those together and put another protein and put another protein, and then you'll get a chain of proteins, and that'll make your microfilament. It's a whole bunch of proteins put together to make a filament. And then you can actually wrap those. Now that you have a long filament made of a bunch of proteins in a line, you can wrap those around each other, like cable. You know, cable can form from wrapping two lines around one another. Like if here's my first protein fiber, then you get a second protein fiber and kind of wrap them around one another, and now you got something a little bit stronger. And that can be a microfilament. And it's made of a bunch of individual proteins bonded together. So we're getting greater and greater structure here. I'll kind of show you. I got a good video that shows this kind of close up. Yeah. Okay, so here's microfilament. So that's a protein, that's a protein, that's a protein. You can see you can put them together in a line, and those can kind of twist around one another. There's actually looks like there's a big protein and a smaller protein. Big protein, small protein. When we start to look at the structure of muscle cells, do you remember muscles made of actin and myosin filaments? Y'all remember that from regular bio?
Mm-hmm.
This is what the actin looks like. So, if you take some of these protein filaments, you put a whole bunch of them together, you can have an intermediate filament. There's a protein filament, there's a protein filament, there's a protein filament. Wind them all together. Now you have something that starts to look like the structure of a rope. If you were to look at a rope up close, you can see a rope is actually made of many, or a cable, like the big cables that hold the bridge up. Y'all know the big bridge there? You can see it's actually, the cable is actually made of smaller cables hooked together to make one bigger cable. Now you've got an intermediate filament. Microtubules are hollow inside. So there's a protein, there's a protein, there's a protein, there's a protein. They hook together to make kind of a circle. And then you put another circle under that, and another circle of them under that, and pretty soon you have a hollow fiber. there's what it looks like if you were to be able to image it closely enough. Here's a better look. The protein is called tubulin here. It makes up a microtubule. And tubulin comes in what we call dimers. Dimer means two proteins. Alpha tubulin and beta tubulin hooked together. So back to my models here, my beads. This might be alpha tubulin. Here's beta tubulin. They're chains of amino acids, but if you fold them up, now you have this tertiary structure. Again, alpha tubulin, beta tubulin, you hook them together. Now you have this, what we call this dimer. Two proteins hooked together in a dimer. And then look how you put them together to make the tube. Takes a bunch of them, you see. And the tube's hollow in the middle. These microtubules move all over the cell. And often things move along the microtubules. You can have the microtubules form almost like a highway. The what? Motocrine tubes? Oh, motor proteins. Okay, so yeah. So here is a microtubule, and here's a motor protein. It's called kinesin. It's a protein that basically can walk. It almost looks like it's two legs walking, carrying a vesicle with it. And so you can see these kinesin proteins will spread out. See how they're spread wide there? And then they're spread thin there? It's almost like they're walking along the microtubule. I got video footage to show you how that looks.
Movements of vesicles, organelles, and the cytoskeleton. Kinesin is a motor protein that moves vesicles and organelles toward the plus ends of microtubules.
See how it's almost like walking along the microtubule? There's the microtubule. Let's see that one more time.
Motor proteins cause movements of vesicles, organelles, and the cytoskeleton. Kinesin is a motor protein that moves vesicles and organelles toward the plus ends of microtubules.
So there's the microtubule there. There's the vesicle. That's a bubble in the cell that's carrying something, and they don't just move around randomly. They're moving using motor proteins. More video footage.
Motor proteins too tiny to see are carried in vesicles, the small spheres, along microtubules, the tracts.
This is called phase contrast microscopy. Whoops.
Motor
Get rid of this voice. So, all of these little things are vesicles in a cell. These lines you see here, those are the microtubule tracts. And you can see how the vesicles move along the tracts. This is just looking up close at one part of a cell. Think about this. This is going on inside your cells right now. Inside every one of them. There's all this activity. And you have 80 trillion cells in your body. Here's another video of it.
The cell allows it to take in the raw materials it needs to stay alive. Cells trap these materials from the outside and package them into tiny cargo bubbles called vesicles. The bubble hitches a ride on a motor no bigger than a molecule, which latches on to one of the miniature monorail tracks that form the cell's transport network. Usually, the bubbles reach their destinations within seconds. But in the biggest cells, which stretch from the spine to the toe, the journey can take 4 days. Each bubble carries a code, a chemical address label that guides it to a particular location. When it reaches its target, perhaps a lysosome, one of the cell's many recycling plants, it hops off the track and empties its cargo. The job of the recycling plant is to break down the delivered raw materials into smaller building blocks, which the cell uses to grow.
See, that's a lysosome, and that was a vesicle dropped off at the lysosome. Okay. This shows the arrangement of the microtubules in flagella. I remember having to memorize all this in college. This is called a 9+2 arrangement. There's 1, 2, 3, 4, 5, 6, 7, 8, 9 sets of tubes in a circle and 2 tubes in the middle. This is taking a flagella like this, cutting it, looking down on the inside of it. That's what it would look like. It has a 9, what we call a 9+2 arrangement. Why does that matter? I don't know. Just that's how it is. You see the same sort of arrangement in these. What are these called? What's this? Centrosomes. These are the centrioles within a centrosome. You see the same kind of arrangement, except it's not a 9+2 arrangement. It's a 9+0 arrangement.
There's nothing in the middle.
Cartier. You're all over it. It's a 9 with nothing in the middle. Here, the flagella are 9+2 arrangement. Perhaps they're linked in the way they form, having similar arrangements. Let's talk a bit about connections between cells and what lies outside of a cell. This is what we call, here's the cell membrane, here's inside the cell, and here's outside the cell. Often the cell has stuff hanging off of it that sticks into its, what we call the extracellular portion outside the cell. And often there are proteins coming off of the cell into this extracellular region. And so the area outside of a cell usually has a bunch of proteins that are sticking off of the cell in it. We call that area the extracellular matrix. Matrix being, a matrix consists of a whole bunch of stuff outside of the cells. So that's the extracellular matrix. Matrix is just kind of a word in biology that means a whole bunch of stuff. You can see there's not only proteins, but there's also carbohydrates out there. Do you know what polysaccharide means?
Yeah.
A polysaccharide is many sugars hooked together in a long chain. Remember monosaccharide, disaccharide, polysaccharide. So sticking off this cell is this polysaccharide chain with a bunch of proteins hanging off of it. See this polysaccharide chain with a bunch of proteins and carbohydrates? Often that's used to help identify cells. A white blood cell might crawl up to this cell and kind of feel it and see if its protein feels right. If this protein feels right, then the white blood cell will leave it alone. It knows it's part of you, and your white blood cell won't attack it. If the white blood cell feels the proteins that are hanging off of the cells, and it feels foreign to them, it'll attack that thing, thinking it's some kind of foreign invader. And it most likely would be. Something in your body that shouldn't be there. This is why you have to get a matching donor when you're getting a transplant. Have you ever heard of that? If I'm going to get somebody else's heart, it has to be a matching donor. They have to have the same types of proteins coming off of the outside of their cells as I do.
Is it the same thing with blood?
Blood types are the same thing. Blood cells have proteins on their surface. They have to match. So if you have type A, you have the same type of proteins on the surface of your blood cells as somebody else who's type A.
Well, type O.
Type O has no proteins on the surface for distinguishing. So anyone can get a type O blood from someone else.
What type O?
You're type O? Most people are type O. About half of people are type O.
So if we have the technology to make vaccines by sticking specific proteins that we want onto a virus, why don't we have the...
That's not really what a vaccine is. In a vaccine, they take the virus that has these foreign proteins on its surface, and they take out the DNA of it so it can't reproduce. And they inject those into you, basically. And then your body recognizes that there's this foreign virus in you, but it can't reproduce, it can't cause sickness.
Okay.
And then your body starts creating cells to kill that particular virus.
Okay. Because I thought I read that they took a virus from a different animal and put proteins on it.
They may have other ways of making viruses that I don't know about. Very well could. I mean of making vaccines. So maybe they're doing something else with the proteins, I don't know. Very well could be. It's a real cool field. And a lot of money in it. Especially right now. Whoever comes up with the vaccines for coronavirus is going to be rich.
I'm going to do it.
They're throwing numbers at it with T's on trillions.
That's what I'm talking about.
This is one of my favorite words in biology is plasmodesmata. A plasmodesma is singular. It's a connection between two plant cells. It allows the cytoplasm of one plant cell to flow into another plant cell. It's a connection between the two cells. It's called a plasmodesma. Many plasmodesma are called plasmodesmata. That's the plural of it. I just love that word. Will you say plasmodesmata?
Plasmodesmata.
Isn't that fun?
Mm-hmm.
It's connections between plant cells. And that's how plant cells just kind of share their material. They kind of stick together in one another, and the stuff just flows from one cell to the other. So they can kind of communicate by sharing molecules. Animal cells don't have plasmodesmata, but they have other types of adjoinments. This is called a tight junction. Doesn't it look like a bunch of rivets somebody shot to join, to join a board to another board? It's like there's a bunch of proteins sticking two cells together. Here's one cell on the left, here's another cell on the right. These tight junctions are proteins that join the two together. And you can imagine those two cells are tied together now. You're not going to get those apart very easily.
Do things pass through those?
What's that?
Do things pass through the proteins that are connected to it?
Say it louder.
Do things pass through?
Oh, pass through the protein?
Yeah.
Not into tight junctions.
Oh.
Nothing can pass through. These are solid. They're like a couple of bolts. And here's another solid. A desmosome is another junction. Here is a kind of a protein. This is the blue is the plasma membrane, and the red is protein, and the green are little smaller proteins that can join one cell to the other. They're kind of stuck together. And the yellow is more proteins on this side, intermediate filament proteins. And so this is almost, I like to think of a desmosome almost like Velcro. You know how Velcro has all those little spines and it sticks together and somehow they kind of hook together and it makes it hard to pull off? That's kind of like this. Doesn't that look a little like Velcro to you? These proteins will stick together, you see, and form a nice little junction. That's called a desmosome. And then finally, this was the one you're talking about, Carter, where you can pass stuff through. These are called gap junctions. Some animal cells have these, where the protein that forms here has a hole in it, and another protein in the other cell has a hole in it, and the holes meet up. So you can actually pass stuff through the cells. That's more like a plasmodesmata. Gap junctions are in animal cells, plasmodesmata are in plant cells, but stuff can pass actually between the cells. Most of the cells in your body are connected to other cells. You don't have a whole lot of free-floating cells. You do have some. Your blood cells just kind of float around, and your white blood cells do too. But most cells, like your skin cells, they're connected to one another. The cells that make up your heart, they're all pretty much connected to one another. And these are some of the connections. You like it?
Oh my gosh, I love this video so much.
This is the best video you'll ever see.
Wait, this is the one
This is the
that
one
shows
with
what?
the motor thing moving.
Yes,
It is
I love
the best
that.
thing that I've ever seen. I actually just
really
googled this
sad.
video like 20 years ago.
I just like randomly remember it.
No,
It's
when you
great.
played the video, the stuff moving, I googled it because I love this video so much.
It's the
Yeah.
best video in the world.
It really is.
It's also got good music.
watch again.
So, these are white blood cells inside a blood vessel, you see. The red blood cells moving along. And this shows how the white blood cells attach themselves to other cells. These are the attachments. That's extracellular matrix. So that's this is the floating stuff you see on the bottom there. That's the lipid bilayer. This is inside the cell. And you see the lines here are all part of the cytoskeleton. And they go into some detail on how it's built and torn down here. That's all cytoskeleton inside the cell. And here they're putting together one of those microfilaments. The cell is. It's assembling it into a structure. And it can also tear it apart. This is an enzyme that tears apart. This is a microtubule being built. Putting it together. And it's microtubule being torn apart. That's a vesicle. There's the motor protein. You see it walking along.
Hell yeah.
Look at your
Pulling that vesicle.
Look at your
There's a mitochondria in there. In the top right hand corner. That's the centrosome with the centrioles. This is RNA coming out of the nucleus and attaching to a ribosome. There's a ribosome, and the ribosome making a protein. There's a little dimer of a ribosome being put together. There's a mitochondria in the background. There's a ribosome squirting a protein into the ER. Leaving. That's the Golgi apparatus in the background and the vesicle being pulled by a motor protein in the foreground. There's the Golgi. Vesicle's going through it. And here we have some exocytosis getting rid of some proteins. This is outside the cell now. That's the extracellular matrix. There's the cell membrane on the bottom. There are the proteins. Proteins can stick out and grab on things. Here they show some of the proteins doing that. Sticking out and grabbing on. What are they grabbing onto? They're grabbing onto the white blood cell that's on top of them. And that's how the white blood cell communicates. It grabs onto the proteins in the cell. White blood cells can actually squeeze between cells in your body. You go investigate other areas. It's all happening right now in your body. If you're wondering how many white blood cells there are, there's about 9,000 white blood cells in a single drop of blood.
That's a lot of white blood
9,000! And how many red blood cells are there? About 5 million.
That's a lot.
In one drop.
Round number.
Lots of cells. Go to your board, assemble a cell. I'm going to come over. For each person, I'm going to point to 3 or 4 things. I want you to say what it is and what it does. And be practicing until I get to you. You can turn